to all the EAPCET aspirants. This is your Prasanna ma'am from NEET World, and yes, today we are going to begin with biological classification, wherein every year of EAPCET paper from this chapter definitely there are 3 questions for sure. Okay, 2 to 3 questions for sure asked from this chapter, and few set of the questionnaire are asked in connection with biological classification and plant kingdom as such. So yes, first let's look into the fundamental insights of biological classification, and then we will go into the theory and EAPCET PYQ practice. So what are the fundamental insights of this chapter? What are the major concepts that we need to cover for the exam? First is kingdom systems of classification. First is scientific method of classification by Aristotle, 2 kingdom system, 3 kingdom, 4 kingdom, 5 kingdom and 6 kingdom system of classification will be covered under kingdom systems of classification. Amongst all the kingdom systems of classification, 5 kingdom system of classification is widely accepted as it has considered evolutionary relationships, which is given by R.H. Whittaker, right? So, we are going to learn in detail of all those 5 kingdoms in detail, their classification, general characteristics with examples. So, for that first is what? Kingdom Monera, then kingdom Protista, kingdom Fungi, Plantae and Animalia. After completion of all these 5 kingdoms, then we will move on to those organisms which are not included under 5 kingdom system of classification. I will tell you the reasons. Those organisms which are not included under 5 kingdom system of classification, like viruses which are infectious agents or living entities, and viroids and prions, and then lichens. These are not included under 5 kingdom system of classification. This will sum up the chapter of biological classification. Each and every segment of this chapter are important, but I am telling you now only. Okay, next. So, let us begin with kingdom system of classification. As I told you, the first scientific method of classification, or who laid the basis for scientific method of classification, is Aristotle. So, first we will look into Aristotle's classification, then 2 kingdom system of classification by Carolus Linnaeus, 3 kingdom system of classification by Ernst Haeckel, 4 kingdom system of classification by Copeland, 5 kingdom system of classification by R.H. Whittaker, 6 kingdom by Carl Woese. Okay. First, Aristotle classification. Aristotle has classified the living organisms based on gross morphological features. Just by looking into the phenotypic characters or phenotypic features, Aristotle has primarily classified the living organisms into Plantae and Animalia. Plants are further classified into herbs, shrubs and trees based on the height of the plants, and then animals based on the presence or absence of RBC in the living cells. They are classified into anemia and anemia. Anemia, those which has RBC; anemia, those which doesn't possess RBC. Okay, anemia and anemia. Okay, this is about Aristotle classification. After this, 2 kingdom system of classification, which is proposed by Carolus Linnaeus. So, the major point that is asked from 2 kingdom system of classification is criteria for classification. What are the major criteria for 2 kingdom system of classification? Criteria includes cell wall, mode of nutrition, response to external stimuli, locomotion, contractile systems. These are the 5 criteria for 2 kingdom system of classification, of which the major criteria for 2 kingdom system of classification is cell wall. Cell wall stands as the major criteria for 2 kingdom system of classification, and the 2 kingdoms that are given by Carolus Linnaeus is what ma? Plantae and Animalia. And as the cell wall is the major criteria, all those organisms, be it prokaryotic, eukaryotic, autotrophic, unicellular, multicellular, all the organisms, if they are possessing cell wall, they are placed under the kingdom Plantae. This is the major point that you have to remember. And all those organisms, be it unicellular, multicellular, autotrophic, heterotrophic, prokaryotic or eukaryotic, if they doesn't possess cell wall, they are placed under the kingdom Animalia. I hope this is clear to you, Nama. That is why cell wall is the major criteria for two kingdom system of classification. Do remember. And then mode of nutrition, Plantae, of course it is autotrophic. Response to external stimuli will be fast in animals because they have prominent nervous system. Locomotion is absent in plants, present in Animalia. Contractile system is present in Animalia and absent in plants. Now, if we have to look into the examples, what are placed under the kingdom Plantae? Bacteria, see, be it a prokaryote, it is having the cell wall, peptidoglycan cell wall. So, bacteria is placed under Plantae. Algae has cell wall. What cell wall? Cellulosic cell wall, or it also has hydrocolloids above that like algin and keratin. Yes, it has cellulosic cell wall, so it is again placed under Plantae. Fungi, chitinaceous cell wall, placed under Plantae. Plants, of course, it has cellulose, hemicellulose, pectins as the composition of its cell wall, and it is placed under Plantae. Now, if you look into Animalia, protozoans, which are unicellular eukaryotes, vertebrates and invertebrates, all of them are placed under Animalia because they doesn't have cell wall. I hope this table is clear to you. If you want, you can take a screenshot also, ma. Listen each and every point clearly, make a note of it keenly. If you are not understanding anyway, you have complete liberty on dropping the messages in chat box and asking me. Okay, because at the end when we are practicing the questions, it should not be difficult for you. I hope this is clear, right, ma? Next, three kingdom system of major thing. Drawbacks of two kingdom system of classification is a major aspect or major segment. In your academy or in your text, there is a paragraph which clearly compares between two kingdom system of classification and five kingdom system of classification, highlighting the drawbacks of two kingdom system of classification. Just now in the previous table, we have seen that be it autotrophic, heterotrophic, eukaryotic, prokaryotic, unicellular, multicellular, they are dumped under the same kingdom, which gave a clumsy classification. This will be the highlights of the drawbacks for two kingdom system of classification. The major drawbacks include prokaryotic bacteria like blue-green algae, like cyanobacteria, blue-green algae, cyanobacteria are placed along with eukaryotes like algae and bryophytes. Next, unicellular organisms like Chlamydomonas, which is unicellular algae, were placed along with multicellular organisms like Spirogyra. And then photosynthetic green algae is placed along with non-photosynthetic fungi, which is multicellular. Okay, so green algae and fungi are placed in the same kingdom, that is Plantae. One is photosynthetic, the other is heterotrophic, which is not photosynthetic. You have to remember the examples for sure. These examples are mentioned in our text, we have to remember it for sure. I hope till here it is clear, right? What are the major criteria of two kingdom system of classification? What are the kingdoms involved in two kingdom system of classification? Who is the scientist who proposed that? And lastly, what are the major drawbacks of two kingdom system of classification? And then here you see Ernst Haeckel. Ernst Haeckel is the scientist who has proposed three kingdom system of classification. So here in three kingdom, four kingdom, five kingdom, one more point add on to the previous things are, what is the new kingdom that is introduced in three kingdom system of classification? Along with Plantae and Animalia, the new kingdom that is introduced is Protista. Okay, Protista is the new kingdom that is included in three kingdom system of classification by Ernst Haeckel. This is the new kingdom that is introduced, which includes all the unicellular organisms primarily. In three kingdom system of classification, Protista is the new kingdom introduced by Ernst Haeckel, wherein all unicellular organisms are placed in it, be it prokaryotic or eukaryotic. Next, in four kingdom system of classification, which is proposed by Copeland, new kingdom that is introduced is kingdom Monera. Here, all the prokaryotic unicellular organisms were placed, and unicellular eukaryotic organisms were placed, unicellular eukaryotic organisms are placed in the kingdom Protista, and Monera here includes all the unicellular prokaryotic organisms, and here this is the new kingdom that is introduced. And then 5 kingdom system of classification is introduced by R.H. Whittaker. R.H. Whittaker has introduced 5 kingdom system of classification. So here the major criteria for 5 kingdom system of classification includes cell structure, body organization, mode of nutrition, reproduction and phylogenetic relationships. All the 5 criteria that are used to classify 5 kingdom system of classification, you have to fix it in your mind. All the 5 criteria. What are they? Cell structure, thallus organization or body organization, mode of nutrition, reproduction and phylogenetic relationships. Here if you see, in the 5 kingdom system of classification, Monera includes all unicellular and prokaryotic organisms. Okay, all unicellular and prokaryotic organisms are placed under the kingdom Monera, and Protista, as we have discussed, all the unicellular eukaryotic organisms, and fungi, multicellular eukaryotic heterotrophic organisms. Animalia, multicellular eukaryotic heterotrophic holozoic mode of nutrition is present. Plantae, multicellular eukaryotic holophytic mode of nutrition. Those organisms were placed under the kingdom Plantae. I hope 5 kingdom system of classification is clear, right ma? Once you look into the criteria of classification, and then we will discuss in detail about 5 kingdom. If you are having any doubts, just drop a message in the chat box. Right, here we go. So this is the table which is given in our text, which is very important. Many a times you were asked with the direct questions from this table itself. But in this table, one minute point that you have to notice is, here cell type, cell wall and a nuclear membrane, 3 of them will come under cell structure only. That first criteria, which is cell structure. Okay, cell type, cell wall, nuclear membrane comes under cell structure itself. Okay, and next is body organization, third will be mode of nutrition. Okay, cell type, cell wall, nuclear membrane, cell structure. Under cell structure these are included. Next is body organization and then mode of nutrition. Okay, phylogenetic relationships were not included under this. Cell structure, body organization, mode of nutrition, reproduction and phylogenetic relationships, right beta? Reproduction and phylogenetic relationships were not included in this table. Few of the students will get confused whether these are the 5 criteria or these are the 5 criteria. Okay, need not to confuse at all. These are the 5 criteria which are mentioned in your text, of which 3 are highlighted in the table. Okay, clear, right ma? Here you see, Monera cell type is what? Prokaryotic. Yeah, and from Protista to Animalia, cell type is completely eukaryotic. Cell type is eukaryotic means nucleus is clearly entitled or uncovered by nuclear envelope. It possesses membrane bound nucleus and membrane bound organelles, and it also possesses ATS ribosomes. These are the key features of a eukaryotic cell. And a prokaryotic cell type includes what beta? 70S ribosomes. Nucleus is not completely uncovered by nuclear membrane. Okay, and then the cytoplasm or the cell doesn't consist membrane bound organelles. These are the key features of a prokaryotic cell. Second is what? Cell wall. Here you see non-cellulosic cell wall, which includes polysaccharides and amino acids, which is nothing but peptidoglycan cell wall. Peptidoglycan cell wall. Clear? And Protista, if you see, in some of the protists, the cell wall is present. Like plant-like protists, chrysophytes, dinoflagellates, the cell wall is present. In animal-like protists, slime wall, cell wall is absent. And here if you see, chitinous cell wall is present in fungi and cellulosic cell wall is present in Plantae. Cell wall is completely absent in Animalia. Nuclear membrane, here you see, nuclear membrane is absent or not completely covered in a prokaryotic cell. Nuclear membrane is present from Protista to Animalia cells. Next is body organization, is cellular body organization, because unicellular organisms are only kept in Monera and Protista. Unicellular eukaryotes, unicellular prokaryotes. And then in fungi if you see, multicellular loose tissue is placed in fungi, and in Plantae if you observe, tissue and organ level of body organization is observed. In Animalia, tissue, organ and organ system level of body organization is noticed. Body organization is again important beta. Next, mode of nutrition. Mode of nutrition if you see, autotrophic mode of nutrition, if you have to consider mode of nutrition, highly diverse mode of nutrition is observed in kingdom Monera. Okay. Highest diversity in nutrition. Diversity in nutrition is observed in the kingdom Monera, okay, because you will observe autotrophic mode of nutrition, heterotrophic mode of nutrition. In autotrophic again you will see photoautotrophic, chemoautotrophic mode of nutrition. Heterotrophic mode of nutrition again has 3 types in it: symbiotic, saprophytic and parasitic. As such, you can see diversified mode of nutrition in the kingdom Monera. Next is Protista, autotrophic and heterotrophic based on plant-like and animal-like protists. In fungi if you see, heterotrophic mode of nutrition, Plantae autotrophic and Animalia is purely heterotrophic again. Clear, right? In this table if you are having any doubts, you can drop a message in the chat box. This table is very important, ma. You will get direct questions from this table, okay. Just fix it in your mind, okay. Here the key points, key takeaways which we have already discussed, look into it. I told you, no, a comparison between 2 kingdom and 5 kingdom system. Monera, all prokaryotic organisms were placed. This though is clear to us. Next, Protista, common ancestor for fungi and Plantae, because Protistans include all unicellular eukaryotes. Unicellular eukaryotes. It consists plant-like, animal-like, fungi-like protists. So they lay as common ancestors for kingdom fungi, Plantae and Animalia. Okay. And then if you see, 5 kingdom system of classification has brought together Chlamydomonas and Chlorella, which are earlier placed in algae, and Paramecium and Amoeba, which are earlier placed in Animalia. In 2 kingdom system of classification, Chlamydomonas and Chlorella, as they possess cell wall, they are placed under the kingdom Plantae. Okay. Paramecium and Amoeba, as they do not possess the cell wall, they are placed under Animalia. And their mode of nutrition is also heterotrophic, but both of them are unicellular, that is why they are placed under the kingdom Protista under 5 kingdom system of classification. Do remember the comparison and explanation properly, so that you will not miss out the statement based or assertion reason questions. Okay. Next, fungi. Kingdom fungi, this is the new kingdom which is introduced. It has again cell wall, chitinous cell wall, multicellular and heterotrophic. Plantae, kingdom Plantae possesses the cell wall and it is purely autotrophic. Okay, it is purely autotrophic. Animalia does not have cell wall and it possesses heterotrophic mode of nutrition. So the brief idea on the 5 kingdoms, I hope it is clear to you. Now we will look into each kingdom in detail. Okay. Before going to each kingdom of 5 kingdom system of classification, we need to know about 3 domain or 6 kingdom system of classification, which is proposed by Carl Woese. Last kingdom system of classification, that is 3 domain 6 kingdom system of classification, once asked as EAPCET PYQ. Now today we are going to exercise this. Okay, this is once asked in EAPCET. Okay. So it is important, ma, look into that clearly. So according to Carl Woese, he said that he has classified the living organisms into 3 domains primarily. Those 3 domains are archaea, bacteria and eukarya, and he has included 6 kingdoms under these 3 domains. Okay. The classification is mainly on the cell wall composition, differences in the cell wall composition, and purely on 16S rRNA. According to Carl Woese, there is a common ancestor for all the three domains. Okay, for archaea, bacteria and eukarya, for all these three domains, there is a common ancestor, and the common ancestor is what? Progenote. Progenote is the common ancestor. This possesses 16S rRNA, which is present in all the organisms of these three domains or 6 kingdoms. Okay, 16S rRNA is present in all the three domains, all the organisms present in these three domains. So, progenote is kept as a common ancestor. Here you see archaea. Cell wall is composed of polysaccharides and proteins. Archaea includes just one kingdom, that is Archaebacteria, and bacteria includes Eubacteria. The rest 4 kingdoms are placed under the domain Eukarya, and those 4 kingdoms include Protista, Fungi, Plantae and Animalia. These are the 4 kingdoms placed in the domain Eukarya. So, here you see cell wall composition in archaea includes polysaccharides and proteins, cell membrane, branched chain lipids. Cell membrane consists of branched chain lipids, cell wall has polysaccharides and proteins, and cell wall of bacteria is composed of polysaccharides and amino acids, which is noted as peptidoglycan. And here you know Protista, Fungi, Plantae, Animalia all possess eukaryotic cells. Clear, right ma? Next. Here we go with each kingdom in detail. I hope kingdom systems of classification is clear for everyone, right? If you are having any doubt, just drop a message. Or give a thumbs up. Yeah. See here, kingdom Monera. Kingdom Monera includes the cells, all those cells which are prokaryotic, and the cell does not possess, there is no nuclear membrane, nucleolus, chromatin, histone proteins are absent, and membrane bound organelles are absent. So, prokaryotic cell does not consist of nuclear membrane, nucleolus, chromatin, histone proteins, it has non-histone proteins. Okay? Membrane bound organelles are purely absent. And if you have to see rigid cell wall is present, and cell wall composition is different in different classes of the Monera. First, if you see in Eubacteria, cell wall is composed of polysaccharides and amino acids, which is peptidoglycan. Archaebacteria possess polysaccharides and sugars, and cell membrane consists of branched chain lipids. Here you see third class of kingdom Monera includes mycoplasma. Mycoplasma are the smallest living cells, we have learnt in cell biology also. Mycoplasma, which is also called as jokers of plant kingdom, this does not have cell wall, there is no cell wall for mycoplasma. Next is actinomycetes. Actinomycetes cell wall composition includes mycolic acid. So, these are 4 domains under the kingdom Monera, and this is their cell wall composition. Important to be noted and remembered for sure. Okay. Next, nucleoid or incipient nucleus. The nucleus which is not completely covered with nuclear membrane is called as what? Incipient nucleus or primitive nucleus or nucleoid. So, here nucleoid is composed of what beta? Double stranded circular DNA and RNA and polyamines. Okay. Double stranded circular DNA, RNA and polyamines. These are the compositions which are present in the nucleoid. Okay. And then you see here ribosomes has what? 70S ribosomes. Ribosomes are of what type? 70S ribosomes. They have high metabolic diversity. Before getting into the ribosomes, you should know the cell wall composition. Bacteria or Eubacteria, cell is completely covered with cell envelope. Cell envelope has 3 layers. We will learn this in detail in cell biology, but for your idea I am briefing it. So, cell envelope has 3 layers. What is it ma? Glycocalyx, cell membrane, glycocalyx, cell wall and cell membrane. These are the three layers of cell envelope. Cell membrane has some extensions called as mesosomes. I will highlight it in the cell biology, and cell wall composition you already know it. And extensions from the cell wall and those structures which are originating from cell membrane includes motile and non-motile structures of the prokaryotic cell. Motile structure includes what? Flagella. Flagella is the only motile structure of the prokaryotic cell which is made up of the protein flagellin. Remember, it is made up of what protein beta? Flagellin protein. And the ribosomes which are present in the prokaryotic cell, they are present inside the cytoplasm, and ribosomes are also attached to the cell membrane. The pure function or the major function of ribosomes is what? Protein synthesis. Protein synthesis is the function of ribosomes. Okay, 70S ribosomes, which is 50S and 30S. These are the two subunits of the 70S ribosomes. Those ribosomes which are present in the cytoplasm are purely involved in what? Protein synthesis. Those ribosomes which are attached to the cell membrane, they are involved in the synthesizing proteins for translocation. Okay, those proteins which are synthesized will be translocated. Next, double stranded circular DNA. Okay, double stranded circular DNA which is placed. This is not completely covered by the nuclear membrane. At the same time, the circular DNA is not scattered in the entire cytoplasm as well. This is placed at a particular location. That is why it is called as what? Incipient nucleus or nucleoid. This has double stranded circular DNA, RNA and polyamines. Polyamines here are positively charged beta. Polyamines will hold the DNA at one location. Polyamines are positively charged, and as we all know, DNA is what? Negatively charged. This is placed at one location as these are binded with polyamines. Next, they exhibit highest metabolic diversity. So when I am explaining you the table, I have told you the highest diversity in mode of nutrition is possessed or exhibited by the kingdom Monera, right? Not just mode of nutrition, even in respiration, kingdom Monera will show highest diversity, okay, highest diversity even in respiration. So both mode of nutrition and respiration together, we will call it as metabolic pathway, right? So highest metabolic diversity also you have to choose the same option, that is Monera. Highest diversity in mode of nutrition also it is kingdom Monera. Highest diversity in respiration also kingdom Monera. Remember that, okay? Excuse. So here in nutrition, as I told you, autotrophic and heterotrophic mode of nutrition. Autotrophic, photosynthetic and chemosynthetic. Heterotrophic mode of nutrition includes saprophytic, symbiotic and parasitic mode of nutrition. Clear, right ma? Highest metabolic diversity, highest diversity in mode of nutrition, highest diversity in respiration is exhibited by which kingdom? It is by the kingdom Monera. Clear, right? Next. Respiration, also it is both aerobic and anaerobic mode of respiration. Again, in aerobic we have two types, one is obligate aerobes, second is facultative aerobes. Obligate aerobes will respire only in the oxygenic conditions. Oxygen is compulsory for obligate mode of obligate aerobes for their respiration. Facultative aerobes, most of the time they act as anaerobes only, sometimes they will act as aerobes, okay. Facultative anaerobes. Facultative anaerobes means most of the time they act as aerobes only. Whenever the bacteria is deprived of oxygen, even in that condition they respire. Even in that condition they respire. Those are what? Facultative anaerobes. Whenever there is absence of oxygen also they will respire. Anaerobic mode of respiration, two types again, obligate anaerobes and facultative aerobes. Obligate anaerobes, completely in anoxygenic conditions only these bacteria will respire. Facultative aerobes, even whenever at few times oxygen is present, even in that condition also they respire. that is facultative aerobes. So here I think you got that metabolic diversity. This is the nutritional diversity that is exhibited by the kingdom Monera, and this is the respiratory diversity, both aerobic, anaerobic, obligate aerobes, facultative aerobes, obligate anaerobes and facultative aerobes. Here you see reproduction. Reproduction is of two modes. One is a sexual mode of reproduction, second is sexual mode of reproduction. Sexual mode of reproduction, you can see gene recombination. Okay, this sexual reproduction, rather calling it as sexual reproduction, it is more evident to call it as gene recombination or sexual recombination. Three modes of sexual recombinations are observed. First is what? Conjugation, second is transformation, third is transduction. Conjugation, transformation and transduction. Next is bacterial appendages. Bacterial appendages, as I told you, the only motile structures in the bacteria are what ma? Fungi. It is made up of what protein? Flagellin protein, and the microtubular arrangement is also important for you. Microtubular arrangement of flagella will be 9+0. 9+0 microtubular arrangement. Non-motile structures include pili and fimbriae. Pili are the tubular structures or the tubular elongations which are the extensions of the cell wall. They help in attachment to the substratum and they also aid in the sexual recombination process of conjugation. How? I will tell you in detail when we are discussing the sexual recombination. And the function that fimbriae exhibits is, fimbriae are nothing but the bristle like structures that are sprouting out of the cell membrane, and they purely help in attachment to the substratum. Attachment to the substratum. Clear, right ma? Next, here you see classification based on the presence or absence of the cell wall and habitat and type of the cell wall. Okay, based on the presence or absence of the cell wall, habitat and type of the cell wall, the kingdom Monera is being classified into 4 subclasses. What are they ma? Archaebacteria, eubacteria, mycoplasma and actinomycetes. You have to remember on what basis the kingdom Monera is being classified. Okay. First is Archaebacteria. Archaebacteria are the most primitive monerans. They are the most primitive monerans. They occur in extreme harsh habitats. Okay, they occur in extreme harsh habitats. So, based on the habitats, like we have halophils, based on the habitat that Archaebacteria is living in, we have 3 classes again. One is halophils, thermoacidophils and methanogens. They are classified into 3 classes again. First is halophils, thermoacidophils and methanogens. So, first halophils you see. Halophils wherein these Archaebacteria live in extreme salty areas which includes halococcus, and thermoacidophils, Archaebacteria which live in hot springs. They tolerate highest temperatures, highest pH and acidic conditions, low pH and high acidic conditions. Okay. So, those bacteria, those Archaebacteria which usually survive in hot springs tolerates high temperature, high acidity and low pH. They are thermoacidophils. Next is methanogens. Methanogens grow in what? Marshy areas. They live in the gut of ruminants. Clear, right? Next. So, the best examples of halophils include Halobacterium and Halococcus. Thermoacidophils include Thermoplasma and Sulfolobus. Methanogens include Methanococcus and Methanobacterium which live in the marshy habitats. Methanogens are present in the gut of ruminants. What is the major role of methanogens? They convert carbon dioxide, methanol and formic acid, they convert it into methane gas. Okay. This methane gas serves as biogas. Okay. This is the production of biogas. How the biogas is produced? From the dung of animals. from the raw substances like carbon dioxide, methanol and formic acid in the gut of the ruminants. Methanogens will act on this and convert this into the product, that is methane. Methane acts as what? Biogas. So, the production of biogas is what? Is from what? From the dung of animals. This is used as what? Biogas. This is the complete description of Archaebacteria beta. I hope Archaebacteria is clear to you. Major thing to be highlighted is the cell wall. Okay, cell wall of Archaebacteria is composed of what? Polysaccharides and sugars. Okay, and cell membrane of Archaebacteria consists of what? Branched chain lipids. Remember that. Whereas Eubacteria cell wall is composed of what? Polysaccharides and amino acids, where we will commonly term it as what? Peptidoglycan. Clear right now? Here you see Eubacteria. Eubacteria are present everywhere. They are cosmopolitan in distribution. So, Eubacteria we have discussed we have discussed cell envelope. It has glycocalyx. It can be capsule or slime layer. Next to cell wall, which is made up of peptidoglycan, that is murin. In Archaebacteria it is pseudomurin. Okay, Archaebacteria it is pseudomurin. Fine, Archaebacteria it is pseudomurin. Here the cell wall is composed of murin, and cell membrane has extensions towards the cytoplasm, that is mesosomes. Mesosomes are purely act as respiratory organs here in the bacteria. Okay, because mesosomes will be releasing out the respiratory substances which will aid in the process of respiration. As such, we will call mesosomes of a prokaryotic cell are equivalent to mitochondria of eukaryotic cell. Remember that beta. Mesosomes of a prokaryotic cell are equivalent to mitochondria of eukaryotic cell. Clear, right? Next, ribosomes. We know the major function of ribosomes is what? Protein synthesis. Flagellum helps in motility. Clear, right? Next. Based on the shapes of bacteria, they are again classified into 4 categories. If the bacteria are spherical, they are termed as what? Cocci. Bacilli are rod-shaped bacteria. Most pathogenic bacteria also. Pathogenic bacteria are bacilli. Spiral shaped bacteria, spirillum. Comma shaped vibrio. Okay. And then metabolic diversity, if you see. Metabolic diversity is maximum in Eubacteria, as we have discussed already. Metabolic diversity includes both anabolism and catabolism. Under anabolism, we will discuss about nutrition, catabolism, respiration. As we have discussed already about nutritional diversity, these are both autotrophic and heterotrophic mode of nutrition. Autotrophic again, photoautotrophic, chemoautotrophic mode of nutrition. Heterotrophic, we have 3 subtypes: saprophytic, symbiotic and parasitic. Let us exercise all the modes of nutrition with examples. Here you see photoautotrophic and chemoautotrophic mode of nutrition. The name itself is saying beta, there is no confusion here between photoautotrophic and chemoautotrophic. The name here itself it is delivering. Photo means what? Sunlight. So, the energy used over here is what? Sunlight. Sunlight energy is used in the form of photons. The energy of sunlight is used in the form of photons. Photoautotrophic, right? And chemoautotrophic, here what energy is used? ATP is the energy used in chemoautotrophic mode of nutrition. Here the electron donors, if it is oxygenic photosynthesis, electron donor will be water. If it is anoxygenic photosynthesis, electron donor will be H2S. And chemoautotrophic mode of nutrition, electron donors are always inorganic electron donors like nitrogen, phosphorus, ferrous, sulfur or hydrogen. Okay, these are the inorganic electron donors. You have to remember for sure what is the energy utilized for photoautotrophic mode of nutrition, what is the energy utilized for chemoautotrophic mode of nutrition, and what are the electron donors both for photoautotrophic and chemoautotrophic mode of nutrition. Energy used and electron donors are very important beta. Connected questions will be asked in connection with photosynthesis in higher plants. See, photosynthesis in higher plants. Okay, in connection with this chapter about electron donors and energy usage, the questions will be asked. Examples under autotrophic mode of nutrition includes chloronema and chlorobium. Chemoautotrophic mode of nutrition, best example we have taken here is nitrifying bacteria, and the major point to be highlighted over here is chemoautotrophic mode of nutrition. Those bacteria which conduct chemoautotrophic mode of nutrition are majorly helpful for recycling nutrients like nitrogen, phosphorus, ferrous, sulfur. Remember that. Now we will look into the nitrifying bacteria for nitrogen fixation. First, ammonia is fixed into nitrates by Nitrosomonas nitrococcus. Nitrates are converted to nitrites by Nitrobacter and Nitrocystis. You have to remember this for sure. N number of times questions have asked on, questions are put on the nitrogen fixation bacteria. Okay, nitrifying bacteria, where which bacteria is involved in converting ammonia to nitrates and which bacteria act on nitrates in converting it to nitrites. Clear, right, ma? Now heterotrophic mode of nutrition. We have three modes here: saprophytic, symbiotic and parasitic. Saprophytic mode of nutrition, here majority of them are what? Heterotrophs. Majority of heterotrophs will conduct saprophytic mode of nutrition only. Here what happens is, primarily the bacteria will release out or it will secrete out the digestive enzymes to the food material or to the substrate which is there in its close proximity. These digestive enzymes or digestive particles will convert the complex insoluble substance into simple soluble compounds, and then those simple soluble compounds are intaked by the bacteria. This is how the saprophytic mode of nutrition will work. Next is symbiotic mode of nutrition. In symbiotic mode of nutrition, what happens, ma? Rhizobium, best example for symbiotic mode of nutrition is Rhizobium leguminosarum bacteria, which is gram negative bacteria, which is symbiotically associated with roots of leguminous plants, whose major function is what? Nitrogen fixation. Major function is nitrogen fixation. Clear, right? Next, parasitic mode of nutrition, which lives on living plants, animals and living organisms. Examples include E. coli and Salmonella. This is about mode of nutrition, both autotrophic and heterotrophic. Next, you see respiration, as we have discussed, aerobic and anaerobic respiration. First, aerobic respiration, obligate aerobes. Obligate aerobes means they respire only in complete oxygenic conditions. Oxygenic conditions. Examples, Bacillus subtilis. Facultative anaerobes. Facultative anaerobes means what? Those bacteria which are completely aerobic, but at some times they act as anaerobes in the absence of oxygen. Examples, Pseudomonas. As such, in the same way, obligate aerobes and facultative aerobes. These are obligate anaerobes and facultative aerobes. What is the term facultative aerobe means? Most of the time it acts like anaerobe only. Okay, whenever oxygen is present, it acts like aerobe as well. So here obligate anaerobe example is what? Clostridium, and facultative aerobe example is Chlorobium. Most of the photosynthetic bacteria will respire as facultative aerobes only. Point to be highlighted and remembered. Most of the photosynthetic bacteria will conduct what mode of respiration, beta? Facultative aerobic mode of respiration. I hope the complete metabolic diversity is clear to you, to everyone. Here we go. Cyanobacteria, blue-green algae, cyanobacteria, which is prokaryotic. Okay, prokaryotic. This is a prokaryotic cell. So, cyanobacteria can be unicellular, colonial or filamentous, but here the model example we have taken is a filamentous cyanobacteria like Animalia or nostoc. Animalia or nostoc. So here you will come across two types of cells. One is vegetative cell and the second is heterocyst. Vegetative cell includes photosystem 1 and photosystem 2, and it has the pigment that is chlorophyll A, and this conducts oxygenic mode of photosynthesis, oxygenic photosynthesis. Then what will be the electron donor beta? Electron donor will be water. Okay. Second type of cell is heterocyst. Heterocysts are those cells which are purely involved in nitrogen fixation. Fine, right? Vegetative cells will have two photosystems, PS1 and PS2, and it has what? Pigment chlorophyll A, and electron donor is water. Here you can see chromatophores, which are the extensions of cell membrane towards the cytoplasm. This chromatophores act as thylakoid membranes and they will possess the pigment chlorophyll A. But then chloroplasts are not present in cyanobacteria, remember. Okay. Chlorophyll A pigment is there, but chloroplast is absent. Chloroplast is not present in cyanobacteria. In cyanobacteria, the extensions of cell membrane towards the cytoplasm, these extensions are called as what? Chromatophores. See, chromatophores. Chromatophores possess the pigment that is chlorophyll A, and the electron donor is water, so it is conducting oxygenic photosynthesis. Next, heterocyst, it has a thick, tough cell wall, thick and tough wall, that is heterocyst, which is for nitrogen fixation. And cytoplasm is distinguished into central colorless centroplasm and peripheral colored chromoplasm. Okay. And here if you see in heterocyst, only photosystem 1 is present, whereas in the vegetative cell both PS1 and PS2 are present. So here only PS1 is present, which generates ATP, and this is used for what? Nitrogen fixation, and it has the enzyme nitrogenase. Nitrogenase is the enzyme which is aiding the process of nitrogen fixation, and the nitrogenase enzyme is active only in the anoxygenic condition. This performs the activity only in anoxygenic conditions. Okay. And next, the membrane of heterocyst is impermeable for oxygen. Okay. So this is what about blue-green algae or cyanobacteria. I hope this is clear to you, Noma. Two cells, vegetative cell and heterocyst. What are the compositions of vegetative cell? What is the function of heterocyst? Where chromatophores are present and how cytoplasm is distinguished? This and all you have to know. Apart from vegetative cell and heterocyst, there are some thick and tough cells in between. They are dead cells called as what? Echinates. They aid in the process of vegetative propagation. Okay, they will fragment these filaments into two fragments and they will aid in the process of vegetative propagation. This is about cyanobacteria or blue-green algae, and this is the theoretical description of it. Once go through. They are unicellular, colonial, filamentous and aquatic or terrestrial algae. Photosynthetic autotrophs, they are most primitive and they conduct what? Oxygenic photosynthesis. They form algal blooms in the polluted water bodies because of eutrophication. Okay. And heterocysts, these are the specialized cells which aid in nitrogen fixation. They are present only in few cyanobacteria. Heterocysts are not present in all the cyanobacteria. They are present only in few cyanobacteria like nostoc and Animalia. Okay. Here you see echinates. What are these? They are thick walled dormant cells in blue-green algae which germinate into new fragments. They aid in the process of fragmentation, as such vegetative propagation. Next is Trichodesmium erythraeum. Trichodesmium erythraeum. Okay. This is the blue-green, this is the cyanobacteria which gives or which produces red color in the Red Sea by releasing out the toxins. By emitting out toxins, this produces red color in the Red Sea. This is a marine cyanobacteria. Remember, Don't get confused between the marine dinoflagellate which produces red color in the Red Sea and between the marine cyanobacteria, that is Trichodesmium erythraeum. Here the cyanobacteria which gives red color of the Red Sea is what ma? Trichodesmium erythraeum. Dinoflagellate which produces red color in the Red Sea or the Mediterranean Sea is what? Goniolax. Remember it accordingly. Okay. Next is reproduction. As we have discussed, two modes of reproduction, asexual and sexual mode of reproduction. Asexual, here you see under favorable conditions, unfavorable conditions, and sexual recombination you can go through conjugation, transformation and transduction. So, as of today we will stop till metabolic diversity, and now it is questionnaire time. In our next session we will discuss about reproduction as well. Now let us check our memory. Yeah. Look into this the question which is asked in NEET in EAPCET 2023. Assertion reason type of question. Look at here, assertion, organisms of Monera having pseudomurine cell walls are producers of biogas. Look into it again, organisms of Monera. It is not of all the organisms present under Monera kingdom. It again has 4 different domains like Archaebacteria, Eubacteria, Mycoplasma, Actinomycetes. So, not all organisms are the producers of biogas. It is only Archaebacteria which is producing the biogas. Okay. In reason, if you see, Archaebacteria present in the gut of ruminant animals like cows and buffaloes will produce the methane. So, here A will be false and R is true, option D, right ma. Next question. Yeah, this you can answer. Just now we have discussed about cyanobacteria. Which of the following show the presence of chromatophores? Presence of chromatophores. Is it a heterotrophic bacteria? No. Presence of chromatophores is observed in what beta? Blue-green algae, that is cyanobacteria, right? So, it is not shown in chemosynthetic autotrophs as well. No, it is not a branched filamentous bacteria. Photosynthetic autotrophic eubacteria, that is blue-green algae. So, here what will be the right option? Option A. Yeah, here we go. This is from Kingdom Systems of Classification. Answer it. The difference between holophytic nutrition and holozoic nutrition. Holophytic nutrition is what? Autotrophic or heterotrophic? It is autotrophic mode. So, underline this. This is not uptake. This also can be eliminated. This, right? Holophytic is autotrophic mode of nutrition. Holozoic is ingestion of solid organic food material. It is not utilization of energy. So, here what will be the right answer ma? Option A. Holophytic is autotrophic nutrition. Holozoic is ingestion of solid organic material. Again a question from cyanobacteria. This is the last question we are solving for today's session. Which of the following structures of blue-green algae are sites of dinitrogen fixation? Dinitrogen fixation, it is heterocyst. These are the specialized structures of BGA which are purely involved in nitrogen fixation. So, in today's session we have completed Kingdom Systems of Classification. In Kingdom Monera we have completed Eubacteria, Cyanobacteria up till metabolic diversity. We are left with reproduction and useful harmful bacteria, Mycoplasma, Actinomycetes, and tomorrow we will finish off the Kingdom Protista as well. Stay tuned. At the end of this session I would like to tell you the major key takeaways of this chapter, and then I would like to remind to all the EAPCET aspirants who are aspiring for EAPCET 2026, these sessions are 100% useful for you. So, just tap on the link of NEET World and just view all the classes. Okay, be it the recorded sessions, at least take the screenshots of the major tables and flowcharts which are provided, which will be highly helpful for you. Okay, so that's all for today's session, beta. Thank you.